Please go ahead.
This a little pointer going. Okay, can everyone see my screen?
Yes.
I guess so. Okay, everyone's on mute. So, again, thank you for the kind introduction, and Tony. And as you mentioned, I just joined. I was became aware of the society at the ACR meeting in April, and is really excited that find people who share the same passion that I have and my company has for doing functional medicine and working with 3D cell-based assays. I had read Tony's paper on functional precision medicine and just use that as a guide for some of the things we're doing. And if you would like to go through sort of a story of how I got involved in this, some work I've done previously in 3D cell models and some of the history of 3D cell models, and talk a little about the microfluidic systems. We call it protein fluidics, but really we're doing microfluidics for 3D cell models for organoids, spheroids, tumoroids. And after that introduction, then go through a couple use cases that we've done with some of the groups we're working with, profiling cancer models, looking at the epithelial mesenchymal transition, and then lastly, an area that I'm really interested in is immuno-oncology. And so using co-cultures of immune cells with cancer models to really further precision oncology with functional assays. And as you'll see, a lot of functional phenotypic assays is what I've been doing in my career, and applying these to this precision medicine and functional precision medicine, I think is really exciting. With further ado, I actually am really interested in history of science. I love this, and I went back and looked at the history of 3D cell models. It really started in the 60s when the 3 Rs was introduced, trying to replace animal models with cell-based models, and really came from the HeLa cells from back in the 50s. You could argue that the first real precision medicine, functional medicine, was Robert Sutherland when he took HeLa cells and put them into a spheroid and started looking at them as a sort of novel model for drug discovery. But I think the real change came through use of stem cells. And so human stem cell induced pluripotent stem cells, and using those to form new models, organoids, spheroids, tumoroids, and treating cancer stem cells as a way to create these 3D cell models. Then in 2000, the government really got involved with the Tox-21 program, the Organ on Chip Tissue Chip Initiative by NIH, and most recently, in the end of 2022, the FDA Modernization Act 2.0, which now allows 3D cell models to be used in support of new drug candidates. My background, as I mentioned, is more in the drug discovery area, developing tools, instrumentations for helping with assay development, the screening, the lead optimization, but always been interested in the translation to clinic. Now, in the drug discovery, 3D cell models are being used throughout the whole process, not as intense as you'd like, but we really seen sort of uptick in use, especially in screening and hit to lead. I'll show you a little bit of data from a group that Scripps Florida, leading the way in that area. But definitely being able to recapitulate the human body in a dish, being able to use that as more predictive assays, I think has been really exciting. And then as we look now into the future, being able to use patient samples from the clinic in the drug discovery process, and then using predictive assays that are developed with those samples, to translate into the clinic, I think this is a really exciting area. And with the Society for Functional Precision Medicine, I see this as a key area where we have people like myself who's more involved in drug discovery, working with the clinical physicians, et cetera, and using sort of a feedback loop to really benefit the drug discovery and creating therapeutics for patients. The use of cells has been many decades, and more recently there's been transition to 3D cell culture. And the advantages of that, if you think about cells in a dish, they're really not a natural setup. You're dealing with cells in two dimensions where you don't have the natural cell-cell interactions. You have the cell-plastic interaction, you have different coatings, et cetera. And so it's really an artificial construct that's been useful, but really doesn't, again, recapitulate the human body. And so animal models have been sort of the method of choice for creating that more physiologic relevant model. But animal models, are costly, and there's a lot of other ethical issues with those. And so replacing those 3D cell models, where you can have the extracellular matrix, you can have the nutrient gradients, you can have the cell-cell interactions, you can look at the drug response in a more, phenotypically representative manner. And so that's a really exciting area. Now, the different type of models, ex vivo models, you get a wide range from spheroids, which are, really just clusters of single cell types, but you can have multicellular spheroids. You can do this in different devices. And then organoids is where you have sort of self-assembly from typically stem cells, where they would differentiate into a organoid-like, which has some of the functions of an organoid in the bottle. And you can do this in dishes, you can do this on chips. And then there's also use of tumor fragments, where you take the biopsy from a patient and you have all the cell types in there and you have the extracellular matrix. You can chop it up into fragments and put that in the dish and do work with that. Another area in here is bioprinting. And so a lot of people have been using cell dispensing techniques to try to create tissues in the dish, tissues in different microfluidics formats. And so there's really a lot of exciting work going in here. But I think it all starts with the patient samples, and starting with the patient derived cell lines that you can make into the spheroids and organoids that can then really recapitulate the response of those patients and provide, those functional readouts that allow you to get that precision medicine. One area that I've published on before is using spheroids, and these are mortalized cell lines. In this case, we're using HCT116 cells, and just show that you can create different size spheroids and being able to then use those in high content imaging. On the top here, we show an example of imaging a spheroid with 3 different stains. First is the nuclear stain. You can use calcium AM as a live cell viability stain, and then ethidium homodimer, which permeates dead cells, in the nuclear stain. And using different types of analysis methods, you can count cells, you can do simple intensity responses, but it's a very well-established technique for looking at live cell and viability response. You can do this then 2D and 3D, and we've been able to show that you get significant differences between 3D and 2D behavior when you look at these cell lines and these spheroids in this manner. A lot of times you'll see higher sensitivity or lower sensitivity for 3D cell models. I think the jury is out on why this is. You might have different penetration into 3D, or you might have more resistance when the cells get together. But I think the key thing is that in 3D, you're doing more relevant assays, and you're getting closer to the human body. Another assay here, which is out of Scripps, Florida, by Tim Spicer. Here he's using patient-derived spheroids, using pancreatic cancer lines, and then 2 human-derived pancreatic lines from pancreatic biopsies. And he looking at here at the formation of the spheres. You can see the mortalized cell lines formed by spheres, but the human-derived ones were more formed organoids, and so they're really not forming a strong 3D structure. He added in the cancer-associated fibroblasts, and then was able to see a very cohesive 3D cell model form over a 72-hour period. And so the introductions of these multicellular into the dish was really critical for him to form a good model for doing the screening. He did this in 1536 well placed, so a real tour de force if anyone's worked at that level of miniaturization. And then using the output here, instead of using imaging, he used cell titer glow 3D viability measurement for ATP. And able to see, first, significant differences between just the cell lines themselves and the cell lines with the fibroblasts. And then also differences between 2D and 3D. In this case, he saw a shift for gemcitabine increasing in sensitivity, higher sensitivity for 2D than 3D. Again, just an example of how you can use patient-derived samples in a high throughput screening manner and get some really important data out to help do drug discovery and further drug development. Well, where we also want to go with it, not just looking at the viability output, but also looking at phenotypic changes and looking at secretion from spheroids. And so here we call this more multifunctional profiling. We're not only looking at the decrease in viability and change in phenotype with those, and this is just star-spawn treatment, but then looking at secretion of VEGF and how that decreases with increasing star-spawn concentration. And this is just a sort of simple example, but you can look for secretions of different cytokines, chemokines, growth factors, as well as metabolites. So there's a wide range of information you can get and to do characterization of how these patient-derived models respond to different treatments. One last example, shifting from spheroids, these are organoids. And so this is a group at Vanderbilt University, Yu Young Choi, and they're very interested in liver cancer and stomach cancer. And so these are gastric organoids. These are mouse models where they've taken the mouse cells and grown them into really hollow spheres, organoids, very elegant systems. And then you can then culture them and look at their response to different treatments. So here she's looking at in the incubation with a MEK inhibitor, and see, as you do this over multiple days, you can see decrease in viability and change in phenotype, and so you can see disintegration of the gastric organoid over time, and then just using sort of the simple viability dyes here. But again, just using the power of using patient samples. In this case, it's mouse, but she's moving into using human samples, human patient samples, and then using this then to look for different mechanisms of action and basic research in that area. So let me shift now to my company and microfluidic assays that we've developed, microfluidic systems. So we really provide a benchtop automation device for doing 3D cell model assays, organoids, and spheroids, ranging from our microfluidic flow chips to a system that does pneumatic control of pressure of fluid movement, and a device that allows you to do high-resolution imaging and supernating collection to really get you that multifunctional data out of your samples. Now, if you look at lab-on-chip technologies that have been developed for the past 20, 30 years, the promise has always been smaller volumes, smaller footprint, being able to do unique things that you can't really do in a multi-well dish, replacing expensive automation. But one of the issues have been a lot of these are complicated to use. They can be very expensive, and a lot of them are single purpose. And so you can do a lot of amazing things with them. But what we want to do is overcome some of these barriers and create an easy-to-use organoid system that can work with a variety of different cell types, different organ types, automates the critical steps in that assay flow, and then is compatible with the other instruments in your lab, your imaging systems, your plate readers, your liquid handling systems. So the features of the system we developed, our Puma system, we have a protected sample chamber. So the organoids are loaded into our flow chips. They fall into the bottom of the well in this little chamber. I'll show you some details of that in the next slide. You do automated media exchanges through the microfluidic devices. And this allows you to set this up, whether you want to do changes every several hours or 24 hours or 48 hours, allows you to schedule that. As you're doing the exchanges, you can also collect the supernatants, and now you can look for time response of different aspects of your cell model, whether it be, again, metabolism, immune response, viability responses, and then allows you to do automated immunofluorescence staining. And I think this is an area we find really powerful because of all the steps involved in doing immunofluorescence staining. There's a lot of loss of your cell models, a lot of pipetting, damage of the cell models, that we can overcome that with our system. Now, the heart of our Puma system is our flow chip. Shown here, it's set up as a multi-well plate, a 384 well plate format. So all the wells are in 4.5 millimeter spacing if you're using any automated liquid handling systems or automated plate reader imaging systems. But instead of being individual wells, these are lanes of samples, sample lanes, where all the wells are connected via microfluidic channels on the bottom of the flow chip. Now, the bottom is a thin COC, so it allows you to do imaging up to 100x oil immersion imaging on these, and so that it makes it incredibly versatile for the type of readouts you want. And then you have this protected sample chamber, which I mentioned. So it's a 1.2 millimeter, so it'll fit most organoids, sort of the 300 to 800 micron size 3D cell models. And then you can do exchanges in and out from the side, so you don't disturb your organoid when you're doing your assays. Now that exchange shown here, after the samples and reagents are loaded, you then have the fluids being pushed in and out by pneumatic pressures. And then the fluid comes in and out the side. Now, you do about a 95% fluid exchange. So again, from immunofluorescence staining, from compound carryover, it's very efficient. You also don't dry out your sample. And so you're not having shear forces over your sample. You're not completely removing the fluid. And so your organoid model will stay nice and secure in this bottom, not drying out, not being perturbed by the fluid exchanges. So when we set up an assay, then it's really just a series of fluid exchanges through the sample well. Or you can take reagents, for example, if you want to do a media exchange, you can add in fresh media. You can set the incubation time for that. If you then wanted to do a compound addition, you can move in a compound of a known concentration, incubate that. You can then collect that compound if you wanted to do downstream analysis of the supernatant. You can do multiple of those, and then at the end, you can then do a staining. So add in a stain buffer. You can wash that, and then at the end of this step, then you're ready to look at your images, your organoids on an imaging system. The way we set this up is pneumatic controls. The pressures are all done through a pneumatic manifold that is microprocessor controlled. You select a protocol that has a series of steps, the transfer steps, and you load that and you run. So it's a very simple interface for the user. So the workflow then is really you would prepare your flow chips with your organoids, your reagents. You then run that in your Puma system where you can do your media exchanges, your compound additions. You can inspect them using imaging. You can inspect them using the supernatants. And then at the end, you can do your automated immunofluorescence staining to look for your endpoint biomarkers, or you can do that on live cells too if you want to watch the change in expression of certain proteins or phenotypes, if you will. So let me then move on to some of the applications we worked on. This first is multifunctional profiling, which I mentioned previously, I think is one of the strengths for doing functional precision medicine. So you want to look at many different phenotypes as you can. You want to look for viability, one of the key assessments, especially for drug discovery, drug development. You want to look for secretion of factors. You want to look for changes in immune response, look for changes in metabolism. And then there's also a need to do biomarker detection, and that's typically done by immunofluorescence staining. And so being able to do all these on the same tumoroid sample, same organoid sample is really powerful, because now you can follow the organoid by organoid. You can look for the heterogeneity in organoid formation. If you think about what drug discovery has done, is they try to make the samples as homogeneous as possible. That way they get the best CVs, they get the tightest assays, windows. But by making everything homogeneous, you're taking out that natural heterogeneity of humans. And so what we really think is looking at organoid by organoid and looking at enough different organoids, you can then capture that heterogeneity, which I think is going to be very critical for precision medicine. Now, this work, we worked with a group at Tulane University, Matt Burrow, and she's been doing work with triple-negative breast cancer patients from the New Orleans community, sort of underserved community, prominently a Black community. His initial work had been with mouse models using explants, and he has a lot of published data there, but he wanted to move from using mouse explants to using a higher throughput and a better model, which was to create organoids in chips. And so you worked with him using a different cell lines. In this case, we're showing work from his 4IC cells, which was a metaplastic breast cancer, resistant to different therapies, including Taxol. And here we are forming organoids in round bottom plates and then transferring them to our flow chips. We did a viability assessment, 4 different compounds, ran that in the Puma flow chips, and then did automated drug treatment and staining of those organoids and imaged them on an image stress micro system. So here you can see the viability drug response. The untreated organoids are on the left, and then increasing treatment shows the loss of viability and the destruction of the organoids, as they fall apart, the phenotypic change, if you will. And so if you looked at then the drug response here, what we saw was toward expected values for romidepsin and trametinib. But when it came to paclitaxel, we saw about a factor 100 higher than you'd expect for immortalized cell lines, MCF7 cells, et cetera. And this was, again, consistent with the patient response, which was resistant to Taxol therapy. And so it's a very nice demonstration of being able to recapitulate the patient response by using their cancer cells drawn from their tumor and then grown up into organoid models and then treated in our Puma flow chips. So in addition to the viability, you said, well, we're now collecting these supernatants, and let's look at different secreted factors. For this, we wanted to work and look at the glycolysis. And so for glycolysis, we looked at the lactate secretion. We've been working with Promega. They have a very sensitive assay, luminescence-based assay for lactate and for glutamate and other metabolites. And what we did was to collect a sample every 3 hours from single organoids. These are the 4IC organoids that we're sitting in our flow chip. And so every 3 hours, we would exchange the fluid, change the media, and then collect those samples, put them into a multi-well plate, and run them using this Promega assay. So here's the results from that. And a little complicated. The color code is the time sample, so from 3 hours to 12 hours, and we collected a baseline sample, which was untreated. And then the bottom axis here is the concentration of the different compounds here, looking at the paclitaxel, romidepsin, and trametinib. And then we have a full change in lactate concentration, so a change from this time 0 and 0 concentration point. So you can definitely see a drug-induced shift in the glycolysis, increase in lactate secretion. At the end of this, then we did the viability stain, and you can see, were the cells still alive, were the tumoroids still viable, and they were. We saw some increase in number of dead cells, as indicated by the red staining here, but for the most part, they were still viable and still undergoing significant glycolysis. So then let me move on to the biomarker detection. So here, what we wanted to do was to use our system to look for different biomarkers and really look at the epithelial mesenchymal transition in these breast cancer cells. So here at the end, we took the samples. We did the full immunofluorescence staining where we did your blocking, your primary antibody, washing, and then using a second antibody and with Hertz to do a nuclear staining. And at the end of this, image them. In this case, we're working with Yokogawa on their CQ1 imaging system. If you think about the value to the customers, you're really reducing number of pipetting steps. It's a hands-off workflow. As they showed before, your samples are in this protected sample chamber, so you're not worried about losing them or damaging with your pipette tips. In here, we wanted to compare the 4IC patient-derived tumoroids to MCS spheroids. And so again, a breast cancer model. MCS spheroids are ER positive, non-aggressive, whereas the patients of 4IC cells were highly aggressive metastatic. And so this is the collection of images here, a lot of data, but on the left, we have E-cadherin, which you see a strong expression of E-cadherin with the MCF7s, but very low E-cadherin 4IC cells. This is an epithelial marker. When we look at Vimentin, you see loss of Vimentin in MCF7, but a strong Vimentin staining in the 4IC models. And then you have the keratin, the Fluoridin, F-actin staining, and Vimentin. So it really shows that a strong difference between these MCF7 and the 4IC and the transition from the epithelial to mesenchymal that we see for the 4IC cells. And so again, an example of the power that you can use for biomarker staining to look for differences between cell types, look for, if you wanted to look for drugs and therapeutics that could perhaps be more effective against the 4IC for these type of breast cancer. It gives you those tools to look at those and look for compounds that might affect these transitions. And in terms of the type of imaging analysis you can do, you can go to very high resolution imaging, it's 40x imaging, and you can see the individual cells. You can see the membrane, cell membrane, the localization of the different stains there. And so, from ability then to get different functional information and use that information, I think this is a really exciting area. And we're really happy with the way our customers have been able to use these flow chips to really further their research. So the last area, again, this is more preliminary data, but I think of area of high interest, and that's doing co-cultures for immuno-oncology and looking at functional response of cancer models as they interact with immune cells. So in this case, we are using cancer spheroids. These are MCF7 spheroids. And we have human PBMCs. We got these from Stem Cell Technologies, activated with their immunocult, and then activated the cells over a 6-day period. You labeled the T cells with a live cell stain, cell tracker deep red. And then the MCF7 spheroids, we labeled those with another live cell tracker green. And so 2 non-toxic live cell stains developed by Invitrogen. They're really nice stains if you want to follow cells over multiple days in imaging. We did the co-culture in our flow chip. And the way we did that is we had the spheroids in the center sample chamber and introduced the activated T cells through one of the microfluidic channels. And so this allows you to do controlled introduction of T cells into the same, this region, same chamber with your spheroid. And once they're in there, they're, as I said, they're protected. So they aren't going to be moved around by the fluid transfers. And you can do different compound additions. You can collect supernatant from them. And so it's a very powerful way to look at co-cultures in a stable format, and allows you to do imaging then of those and follow those over time. So here shows transmitted light as it's feeding. You see the immune cells are speckled here, and then the cancer model, the MCS7 spheroid in the center. Again, from a dimension standpoint, this well here is 1.2 millimeters. And so this is probably about 300 micron a spheroid, with several thousand immune cells dispersed through the well. After a 4-hour incubation, you can start to see the immune cells in magenta infiltrate and interact with the cancer model, the MCS7. This is a little 3D rendering created by the Yokogawa CQ1 software, but really showing you the ability now to track individual cells. If you did a time-lapse study, you could watch them move from the well into the tumor, and then ultimately what you're interested in is, do these kill the tumor model? And we see that. So after 48 hours of incubation, here in the green again is your MCS7 spheroid. You can see how the T cells only and spheroid only stay fairly well intact and viable over that 48-hour period. But when you introduce the T cells to the spheroid model, you see disintegration of it, you see loss of viability, and phenotypic changes, so functional change of that tumor model in response to the immune cells. And so then be able to use this as a way to look at different therapeutics that can increase interaction, or if you have a tumor model, a patient-derived model, now resistant to the immunotherapy, how do you overcome that resistance? And so that's the way the groups we're working with are taking this, and really the future for looking at immuno-oncology, again, in a functional manner. By going to high resolution, you can now look at the interactions. And so this is a 40x slice through here. In this case, we fixed the cells and did a staining for CD3 for the T cells. And you can see the different phenotypes of the nucleus as they get sort of striated, that indicates a dead cell. And then the more uniform diffuse nucleus would be a live cell. And here we can see the interaction of immune cell with the cancer cell as it goes and attacks and kills that cancer cell, sort of capture a slice in time. And so very excited about this. And again, being able to follow this on a cell-by-cell basis is, I think, very exciting, and being able to use this as a tool to help guide therapeutic development. And ultimately, if you can take a patient biopsy with the patient immune cells, and then as a clinician be able to use a tool like this to say, okay, here are the different immunotherapy regimes available to you. These are the ones that are going to work best for you. Or to see immune resistance and watch over time as that patient's biopsy, patient's tumor cells develop resistance to different immunotherapies. And so very excited about where this is going. So, I'd like to acknowledge, the people who have involved with it, the team at Protein Fluidics, the group at Yokogawa, which has been helping both with the imaging and image analysis, Tulane University with Matt Burrow and his group, the people at Greiner who provided some support with the assay development and consumables, and then Molecular Devices, Oksana Tsurenko, who helped with the imaging and some of the earlier publications, and her team there. So as a summary, really just want to, the 3D cell models, I hope, is able to, show and convince people are really critical to advancement of functional precision medicine and drug development. What we've done at my company is develop this benchtop system for automating some of the, sort of standard methods used and improving the efficiency of those, using 3D cell models to do media exchanges, immunofluorescence staining, supernatant collection, secretion analysis. And really, I think the immunofluorescence staining is one of the key areas that's going to allow to get a deep dive into these models and how they're performing the functional changes in them. And gets you this comprehensive phenotypic data set that ultimately can be used, with artificial intelligence, machine learning to try to pull out some, response scores or try to qualify how different patients would respond to therapeutics. But ultimately, it's the patient-derived samples, patient-derived organoids that are going to provide that information, and in the future are going to be used for a wide range of things, including disease modeling, drug discovery, and precision medicine. So with that, I'll thank you and be happy to take any questions.
Great, Evan, that was really excellent. I think we all have to realize in our field that the FFPM field really is we're studying the art of the possible, and these like important technological, or say, advances that allow us to do anything like easier, cheaper, faster, fewer cells are really going to be critical for us to move forward. There's several questions. I'm going to take my prerogative and ask one. One thing I wasn't clear on is the immunofluorescent imaging actually built into that sort of shoebox size device, or do you remove the chip from it to a fluorescent imager?
So currently the setup that you would remove the chip and put it into the imaging system. That's sort of like this shown here. So we've set it up in a multi-well plate format, so it's compatible with any high-con imaging system. But I've developed and sold imaging systems in the past, and they're very expensive, long time developed. So for here, we wanted to allow our users to take advantage of their current imaging systems and not overcomplicate our system. Having said that, having imaging in there so you can watch it in real time is going to be very powerful and something we're definitely looking at.
Got it. That was my next question, the real-time imaging aspect.
Yeah.
I think I'll turn to the audience. What I'll do is I'll try to see if we can, as people have just asked their own questions rather than have me ask it. Now, Sandy has a couple questions. Sandy, can we start with you? Could you unmute and just directly ask your question to Evan?
Sure, a few questions. First, in the screening device, can you control the temperature, control the partial pressure of different gases? In particular, I'm concerned about oxygen.
Yes, we can. We have just launched a new system. We call it our Puma System EC, which allows you to do environmental control. And so it keeps the sample at 37 degrees or 5 degrees above ambient up to 40 C. You can't go too high because our plastics will start melting, but that's an aside. It allows you CO2 control. And there's also an option for purging with nitrogen, so you can control oxygen levels.
Okay. And then one thing you sort of touched on after I'd asked the question, I wasn't quite sure of the full answer. Let's say you have a mixed population of cells in a particular organoid. Can you use distinct markers and follow over time the differential sensitivity of the two cells to the particular drug in there?
So you can use different markers. I mean, there's different live cell stains. For example, here these are two different cell tracker stains, and so as the cells die, if you're looking at viability and differential viability, you would see the cells go away and reduce the number of cell count. If you want to look for differential expressions of biomarkers, there, once you fix the cell, you sort of have a slice in time. So you definitely have that possibility. I think the other way people are working is, they'll create cell lines that are expressing different proteins, expressing RFP, GFPs, et cetera. And so then you can use that as a tool to look for, response of different cell types within a multicellular structure.
Great, thanks, Sandy. We have a question from Thomas. Is he available? Thomas, are you available to ask your question?
Hi there. Sorry, I was in the lab, so
all right.
I'm still very much working in the lab. Thank you very much for a great talk.
Sure.
I was wondering about hydrogels. I think you mentioned that you've used Matrigel, specifically as well with the immune cell infiltration. Is that while hydrogel is present in that sort of culture well, or is there no hydrogel present?
So in this case here, we do have hydrogels present in the well. What we find is we use a reduced amount of hydrogel, so about 25% concentration, which is enough to support the organoids over the course of the assay. 2-3 day assay, they'll find and that type of thing. And at 25%, hydrogel will also flow through our microfluidic channels. And so that's one of the big considerations is, too much viscosity can't flow through the channels. Having said that, we've also done work, if you imagine, in the here, you can put a higher concentration of hydrogel just in this bottom chamber, so about 2 microliters. So you can, it sounds like a small amount, but you with practice, it's fairly easy to dispense 2 microliters in here. And so we've done up to 80% hydrogels in here. We've grown organoids in this area, and then subjected them to different treatments. You can flow immune cells in there, and immune cells, they'll work their way through the hydrogels, etc. So there's a lot of different ways you can use this. But bottom line is, yes, we can do hydrogels. We've done different organoids using hydrogels very successfully.
Thank you very much.
Thanks. There's a question from Hinko Gierman. Is Hinko available to ask his or her question? I'm afraid I don't remember Hinko.
No worries, Tony. Yes, I'm here. So the question I had, Evan, great presentation, is on the work you were showing with the T cells. And I was curious if you've been able to replicate that with actually real tumor fragments that already have the infiltrated T cells in there, because it's rather artificial, obviously, to add thousands of T cells. Most fragments would not have that amount. And CD3, CD28 is also a very strong positive activator. So I was wondering if you've done it with native T cells and with anti-PD-1 rather than CD3, CD28.
Yeah, so we've not done that in our application lab. We're a small team, limited resources, but we have a group at Stanford that's working in that area. They've just got it started working in liver cancer. She's MD, PhD, so she has access to liver biopsies. So she's very interested in that. We're also working with a couple of groups in the Netherlands in that area, also looking at
the Volbil Daniela Tallman.
This is a group at Erasmus Medical Center.
Okay, different one. Yes. Okay, great. Very cool. Thank you.
So hopefully in the next 6-12 months they'll get some data out there in publications. So.
I don't see any questions in the chat. I'm going to ask one of my own. If anyone has any additional questions, please put them in the chat before we wrap up. One is that one of the costs, it appears to me, of going from 2D to 3D is many 2D cultures are very simple. You just sort of put them down, they adhere, and you can study your thing really fast and get away. So there's a dimension that, sort of the fourth dimension, that sometimes is overlooked, is the dimension of time. So how long does it take to go from a patient biopsy to a spheroid, and how much time is that adding on to what you might get out of like a one-day 2D culture?
So the short answer is it depends on the sample. So some patient biopsies, you can take them, put them into fragments, and within probably 3 to 5 days, you can have a viable sample to run. Others might take longer time periods, to really form, a good number of 3D cell models where you could perform an assay, have enough, samples to run an assay. It really depends. It's so much dependent on the cancer type, the cancer subtype, but also other disease areas. So, the ones, for example, that I was showing, here from Tim Spicer, this is a formation over a 72-hour period. But, getting the samples to that point is, 1 or 2 month process.
Okay.
Right. So they're taking the patient biopsies. They're finding the cancer cells that are going to propagate. might be, 20% of the cells, and then allowing those to propagate up to the point where they have enough cells then to start creating this spheres.
So can you also comment on like what your take rate is? How often do you get a solid tumor sample that just doesn't progress to forming a 3D culture or organoid or spheroid?
I remember at ACR, the take rate for explants I thought was about 30% or so. A lot of groups will use explants as the model to expand the cells, and then use the explants then form into organoids, tumoroids. So for example, Matt Burrow at Tulane, that's sort of his workflow. And then, but the take rate for patient-derived biopsy and forming those into usable samples, I'd have to imagine is probably similar.
Okay.
Great. Yeah, but that's an area of people are really actively working on. How can you improve that? Because only as a clinician, you say, I can take my patient biopsy and give you an answer. Not I can take your sample and maybe 20% chance I can give you an answer.
Right. Yeah, I think you're right. That is an important technical hurdle. Great. I don't see any more questions in the chat. So I think I will just, for the rest of the audience, thank you for a very interesting description of what your company does. And that concludes this session. I'll remind everyone, August, we are taking a holiday. There will be no SFPM monthly seminar, and we'll resume in September. So everyone have a very nice summer. Take care.
Thank you, Tony.
All right. Thanks, Evan.
Thank you, Tony.